Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing you another really awesome guest involved in work that's helping to create a better tomorrow for all of us. We have the honor today of being joined by Dr. Josh Denny, who is the Chief Executive Officer of the National Institutes of Health All of Us Research Program, which is a really wonderful initiative that ultimately seeks to enroll at least 1 million diverse participants to build a really indispensable resource that helps accelerate precision medicine for all populations. Dr. Denny has been involved since the program's inception. He was originally a member of the NIH Precision Medicine Initiative Working Group, of the advisory committee to the director there, helping to develop the program's initial scientific blueprint. He then led the program's initial prototyping project and the All of Us Data and Research Center, and then ultimately was named CEO of All of Us in January of 2020. Before joining NIH, Dr. Denny was a practicing physician, held several leadership positions at Vanderbilt University Medical Center, including as professor of biomedical informatics and medicine, as founding director of their Center for Precision Medicine and vice president for personalized medicine. And at Vanderbilt, he helped lead discovery and implementation projects in precision medicine, including really interesting work in clinical pharmacogenomics, in building Vanderbilt's DEA Biobank. He was also a pioneer in the area of the use of electronic health records for genomic studies, including the initial descriptions of so-called phenome-wide association studies, which we'll be getting into in a little bit. Dr. Denny is an elected member of the National Academy of Medicine, the American Society for Clinical Investigation, and the American College of Medical Informatics. Did both his master's degree in biomedical informatics and his medical degree at Vanderbilt University School of Medicine. A lot of really important and interesting themes to get into today. We're honored to have him. Dr. Josh Denny, thank you so much for taking the time to come on the show today.
Thank you so much, Ira. It's a real pleasure to join you. I really appreciate your interest and the chance to talk about all of us and the importance of personalized medicine.
Absolutely. Most definitely. Before we get into all of us, would really like to start off with you because you've had a quite an amazing ride over the last several years. Would love to go back to the beginning though and sort of start off with who you are, a little bit of your background, and sort of your interest in obviously informatics and medicine. But I also enjoyed reading that before pharmacogenomics and single and nucleotide polymorphisms and everything that we're going to be getting into, you first sort of developed this really interesting connection between medicine and informatics back in medical school, where you spent some time digitizing sort of medical school training back in the day. And I would love if, as we get into the early days, we could talk a little bit about the Knowledge Map project, which I think was sort of your first foray into sort of this very interesting intersection.
It really was. Thank you. So, starting, I guess at the beginning, I grew up in Kentucky, Louisville, Kentucky. And one of the real narratives in my life was, I guess everyone grows up from a background. And for me, my father had 4 siblings that died of cystic fibrosis at a very early age. So no one in my family was medical until me. But as I grew up with that and that story, it was very prevalent in my life. And I've always been interested in science and medicine. And as I was going through, I guess, middle school even, and then high school, very interested in computers and computer science. Kind of a very different world at the time, but we were able to, at our high school, win through a competition a T1 line. So we actually had, a internet connection and could like get our exposure to email as we started to think about actually some early, worked on some early stuff, even with how you could use some neural nets to play Othello with some of the other high school students. And at that point, the human could still beat the computer pretty easily on those kinds of things. But at the time, I was doing more basic biology research at the time, but always kind of had this drive to how I could do something, and wanted to get into medicine because I felt like there were empty problems and that we could find solutions for. And so I went to undergrad and did molecular biology. That was at Vanderbilt. Did kind of computer science stuff on the side for fun, and then went to medical school. And as I was going through medical school, I was doing more basic research, but it wasn't quite, it was very informative and interesting, but didn't quite energize me quite as much as some other things. And then I hit, my third year of medical school. We had been just, I have this photo of all the documents that we were given. By the first 3 years of medical school, I had a stack that was essentially as tall as I was of notes that I had been handed from my professors and the notes I had taken. And this was not accessible, and there was no way to find this kind of content. There was no Blackboard or WebCT at the time. There's no website for any of this material. We just had all this paper. And so and then people were asking questions like, because I was on the curriculum committee, where do we teach X? Where do we teach, genetics, for instance? And so they get together all of these people and it's like, well, Dr. So-and-so, I think they might teach this. And there's hundreds of people that could be teaching things. And they try to get them together and send out emails and all this kind of stuff. Well, we had the idea, you get to Knowledge Map, we had the idea of, why couldn't we digitize this and collect all these things and make them more accessible? But couldn't we go the step further and actually take that natural language content, and this is in 2000, and map it to some sort of, natural language understanding, some sort of controlled vocabulary that we could tell that it's talking about, congestive heart failure. It's talking about antibiotics, not just a drug, but that drug happens to be an antibiotic, and that you could, map the curriculum. And so that was our task. We did things like we bought, a scanner with an automated sheet feeder and massively OCR'd, hundreds of pages of text. We got, because some of these things were typed and not in a word processing file, we got all the Word files and WordPerfect back then and all these things and digitized them and created a repository. But one of the real innovations was building software to digitize and kind of comprehend in some way, if you will, the content that was there. And so that question of where do you teach genetics is a simple query, and it explodes to things like Watson and Crick and DNA and exons and introns and all these buzzwords that are part of this are all conceptually related. And you can do that query and find that content. And it just kept leading to other things. I did my clinical rotations and I said, gosh, we're seeing all these clinical observations. And then I'm going and reading the literature to find out, what we should do with certain conditions, which makes sense. But you rapidly find that you can be in scenarios in which there's really not any literature out there. I thought, gosh, we're seeing, we have more at that time, more than 1 million outpatient visits a year. Couldn't we learn something from those observations? Couldn't we get smarter from all the contact we're having? There was a great quote around this time, and maybe a little bit later, then HHS Secretary Tommy Thompson said, my grocery store is more advanced than my hospital. And that was basically true. We had incredible systems, but in terms of, we only looked one patient at a time. Electronic health records were brilliant at looking at and saying what one patient had, but didn't give us the ability to look across patients. Yep. And so that was one of my essential challenges. I was like, gosh, we should do something here. And so we started doing the same kinds of things and thinking, well, let's start looking across patients. And I remember a particular patient that we saw in the ICU that had a very odd, unusual eye finding. And you go to literature, the last paper on it was written in 1971. And we're like, well, we've got to be able to know something more about this. So we found, by searching the entire medical record and all the notes that had been written in our patients at Vanderbilt, 117 people that had this condition. And we found that they had, about 40% of them died within 30 days. So, and then we were able to find some of the other associations with that. And that was kind of one of those really compelling examples to me, is like, there's something here. There's something we can do with this and discover that's being missed. Mm-hmm. And so maybe I'll stop there, because it's the start, it's the end of the beginning of what we launched into with what we can do across the electronic health record, which has really been the compelling story for how I've organized a research direction for my career.
Mm-hmm. Yeah. You bring it perfectly up to sort of the next little point on sort of on the timeline here, which as you're describing, we're putting together this tremendous amount of genetic information. You mentioned the electronic health records. The magic that you're performing, the linkages, the connections, understanding how to bring not just one patient, but millions together. A lot of this culminates in 2010 in what, is really described as, a pretty seminal paper that you're involved in here. You lead in Bioinformatics in May of that year, entitled PheWAS, Demonstrating the Feasibility of Phenome-Wide Scan to Discover Gene Disease Associations, which basically sets the stage for this reverse genetics approach, as it's sort of defined, that, we can had all these genes. We know this is genetic stuff, but you're starting with the health records. Wow, we can look at diseases now for the first time. Oh, and make all these very interesting associations to all the single nucleotide polymorphisms, and once again make these reported disease associations. Take us into 2010, because I think this is an underappreciated piece of the phenome stuff. Reintroduce us to the phenome. I don't know if you saw the episode a couple of weeks ago. We had Lee Hood on talking a little bit about his work in sort of systems biology and phenomics. But take us to 2010 in your work, because this is extremely seminal to this whole piece of the story.
Well, thank you. We had building up to this, we had started a DNA biobank that used, building on the same principles, used people that had clinical blood testing and asked them, instead of just throwing away that blood, could we keep and extract DNA from that blood for research? Yep. And that was the basic consent process. And then we would use, as the sole source of phenotypic information, electronic health record data. And that really had never been done before. And so we started with the collection and building of this biobank, which we call BioView, with those, and that started in 2007, building up those samples. And we had the DNA information, we had the electronic health record information and DNA. And the first questions everyone wanted to ask were things like, hey, let's make sure that we can find signals that were just being discovered at the time between things like type 2 diabetes and genetics. We were just at the very forefront of discovering some of these kinds of things, none of them being done with the electronic health record. But, the idea was in our heads because at this point, people were talking about genome-wide association studies. Right. And for the first time, we were for a couple of years into this idea that instead of I have been carefully doing pipettes and studying this gene for my whole life, and I think it's involved in something. I'm going to look under this lamppost. I'm going to look across the entire genome and see if I can find things associated. And people were really finding things that had never been seen before in that, and that was incredibly powerful. So I was thinking, we have the entire medical record. Instead of just having asked someone whether they have diabetes or not, we have clinicians that have followed these patients and been trying to assess them for anything that's clinically important. So the observation was, could we do the analogous story with a phenome? Yeah. And the beauty of the electronic health records is it's not the questions I thought to ask you, it's what you present with. And so it's your life story, it's your health story that we collect across all these patients. And my work in things like Knowledge Map and its successive applications, I understood how the medical record worked. I knew its organizational database structure and all the coding structures of this. And obviously, I was a clinician at the time as well. And so we created this architecture. Everyone says billing codes are messy, but I was like, this is a good place to start, and I'm going to organize the billing codes. So I sat down with Excel, basically imported the 1,000 billing codes and created a crude hierarchy of 700 plus diseases that were, encoded by these 18,000 billing codes, and created my own mapping. There's all sorts of goofiness in the billing code system, like that creates error and pulls up, tuberculosis and a whole bunch of different places, parts of the billing code, but they're all tuberculosis. So we pulled those together, for instance, for TB, and did the same thing for diabetes and all these kinds of things. And so with this PheWAS paper, we phenome-wide association study, we picked a handful of 5 SNPs that we knew should have associations, and we were able to replicate most of the known associations and picked up a couple of new ones. And so we showed that this kind of thing was feasible to do. It was very fast, and for the first time had looked across diseases in an unbiased, systematic way, like we were doing in the genome. It took a while for lots of EHR-based data sets to become available. We were really one of the first, but over time, it's just become routine. People would do PheWAS all the time. Drug companies use PheWAS as a routine way to screen and de-risk advance potential drug targets in large biobanks. It is a complement to most GWASs to figure out what else that gene could be associated with. And it's just been routine. So it's really taken off and really all across the world, because we've since grown it to map in, the newer coding systems and international coding systems, and continue to sort of grow it as a use case.
Excellent. And just as a side note for those listening and watching, take a look at Dr. Denny's sort of PubMed. A search on Dr. Denny when you have a chance, because I neglected to mention that you have literally hundreds of papers in the peer-reviewed literature on these topics. I'm just going to be talking about a few of them today with you, but it really is an exceptional amount of publication. But leapfrogging now forward to 5 more years to March of 2015. The NIH announces the, as mentioned, Precision Medicine Initiative Working Group. Needless to say, they pull you onto this because of your expertise amongst some other luminaries there. Talk about sort of what's going on in 2015, because this is sort of the genesis of then what comes next in terms of all of us. I did have, there's a couple, about 3 years ago now, I did have the chance to talk to Chris Lunt back in the day, sort of on the technological side, but talk about what was happening in terms of, obviously now you bringing the knowledge about GWAS, PheWAS, and sort of the pre-creation of what will become all of us in a couple of years.
Yeah. So at this point, we at Vanderbilt, a few other sites, a handful of sites have started to implement clinically pharmacogenetics. Research genetics has really exploded in genome-wide association studies. And you have the UK Biobank, which has really started to make an international scene. In 2015, they had just recently released their genomic data set, and that was genotyping arrays across 500,000 people, I believe. I think they'd gotten to 500,000 by 2015. And so people were doing in a large biobank setting, really kind of for the first time that was accessible across the world, these beginning to do these genotype phenotype correlation studies. So at that point, most research studies were very much, if you think about things like the Framingham heart study, which has been so important to discovery of lots of things like importance of tobacco and exercise and those kinds of things, cholesterol, of course. Those kinds of studies are, in the UK Biobank, are you enroll, you create a resource, and people leverage that resource to discover, but you don't really find out a whole lot from that study. There's not much return of information. So there are a couple things put together. So we were thinking, what could we do nationally? The US is a fragmented healthcare system. It's a fragmented research infrastructure, but lots of incredible science happens. We were beginning to think, if we really want to bridge some of these gaps with participants for a long time, we want to think of them as participants, as partners. How can we return value? Computing and cloud computing is just emerging. And electronic health records have really seen a lot of growth across the United States. So there's a lot more infrastructure for electronic health records. And things like Fitbit are becoming more common. And so all these things came together and really create a right ground to think about how we could design a new research study. At this group, we talked about a lot of capabilities. Do we want to just take all these existing cohorts that places like Vanderbilt and Geisinger and other places are starting to build and put them together? Do we, do we want to, there's lots of different capabilities and ways. Do we want to do lots of surveys? Do we want to have lots of deep, in-depth visits with people where we put them in metabolic chambers or whatever? How do we want to phenotype? How do we want to genotype? And so we decided that we wanted to be large. We wanted to have scale. There was an emerging understanding that genetics were non-diverse. And actually, believe it or not, it's actually gotten worse since our first observations, obviously until programs like All of Us are coming on board. More and more genetics were running towards European, and we still have more than 90% of our genetic studies worldwide are based on people of European ancestry. So we thought correctly at the time that we really wanted to focus on diversity, because that was a hole in a lot of clinical studies and genetic studies as well. We wanted to leverage the electronic health record. We believed that was a really important part of telling the story. And, it wasn't universally accepted at the time. I'll tell you, we had a ton of, my team, the Vanderbilt teams and some other networks had a lot of history of this. But there are a lot of people, it was still kind of a new story to talk about using electronic health records as a key part of what we're doing. There's a lot of Fitbits were exciting and other kinds of digital health technologies, but relatively untested. And we thought that would be a really important part of a cornerstone of what we'd go forward with. So we built this plan. We decided that it needed to be a new recruitment. We wanted to be national. We wanted to be focused on diversity. We wanted whole genomes, not just genotyping arrays, looking at locations. We really thought rare variation was going to be important to answer new questions and to find out what we don't know. And, we wanted to leverage what we could get with electronic health records. We didn't want to stop there. We wanted to pull in those digital health technologies. We wanted to be able to ask surveys, get lots of biospecimens, do reassessment over time, all these kinds of things as visions for the program.
And you have a lot of products coming out. I mean, a lot of findings, even after just a few years coming out of what you're up to. And I thought, for the sake of, again, there's a lot out there in the literature. I just pulled a few out, but again, for those that want to go off and check some of this stuff out, go into PubMed and look up some of these papers. I thought, Josh, a really interesting example case to start with would be your 2022 JAMA Oncology paper, which is entitled Association of Pathogenic Variants in Hereditary Cancer Genes with Multiple Diseases. And the one facet, I mean, aside from the fact that, there's a couple 100,000 participants that you looked at here, when it comes to cancer and oncology, and, we normally think about, all these different imbalances. We have a certain gene mutation that's responsible for different cancers, a certain cancer that has many different mutations, what's a driver, what's a passenger, and so forth. In this paper, you also talk about all the non-neoplastic conditions that come along with this. I found that extremely interesting because, we hear so much about the importance of early stage prevention, catching the first cancer cell. And it's more than just, ovarian cancer and colon cancer. If we could also identify connections with ovarian cysts and pancreatitis and all these other precancerous conditions, there's a major benefit that comes from this. Talk a little bit about this particular study, if you would.
Certainly. Great introduction to it, too. In this study, what we did is we took 3 populations. This predates all of us having genomic information in terms of our doing of this analysis. So we included 2 Vanderbilt cohorts, 1 a research cohort, 1 a clinical cohort. So we got people that had clinical genomic testing, and then we pulled in the UK Biobank. And one of the things I want to just highlight there is it gets to a problem that is sometimes underappreciated, but which we are really making incredible strides on, which is combining data across different data sets. Yeah. You have, how do people think about and bill or code people having certain diagnoses in the United Kingdom, for instance, versus, what actually happens in the clinical versus a research. And so we were able to put this together through things like the PheWAS approach, which maps all those things together. There are other things that are even broader that we're employing in all of us to kind of help pull those data together as common data models. And so we put these 3 cohorts together to ask the question for these 23 hereditary cancer genes, which are genes that are being incidentally returned now and are being thought about by clinicians in they see a patient. Most common would be people think about BRCA1 and BRCA2. There are other ones that get to other familial cancer syndromes like Lynch syndrome, which is a colorectal cancer, and other cancer syndromes. And so those cancers are things that those genes are ones we wanted to study to see what other cancers and what other conditions might be associated with this. So we used a PheWAS approach. We pulled together the many pathogenic variants that we might see in these genes and grouped them together and said, if you happen to have a pathogenic variant in one of these genes, what conditions is that associated with? Some of these associations don't have tremendous literature behind them. Right. We know the breast cancer story with BRCA1 and 2 incredibly well. And then there's a list, though, of other things when you start getting these cancer syndromes that might be associated. So in this case, for instance, there were 38 across these 23 genes, 38 really established gene cancer associations. And in these cohorts, we saw them all. They all were very strongly associated. But then we actually were able to replicate 70% of all the conditions beyond cancer and beyond in one study that have been reported as strongly associated in the literature. So that provides a kind of evidence that, it really helps understand things like the prevalence and occurrence of these kinds of conditions as well. And then, like you said, we discovered a number of new associations. 7 were cancers, and 12 were non-cancer associations, like you said, pancreatitis and ovarian cysts. And because they're in the electronic health record too, we can go back and see that, do these people with ovarian cysts and BRCA2 variants, does it look like they just, maybe had ovarian cancer or were they in enhanced screening? And we can start to tease those things out because we have those longitudinal records. We were able to look at the timing and did the doctor know about this need for enhanced screening and see, no, it looks like these are really new associations. And things like understanding that pancreatitis and gastritis and these other associations with the syndromes helps people be screened better in clinical practice. It also helps a provider potentially think of the condition better. If they have the full cohort, we work as clinicians, we work as pattern recognizing machines. And when we see a person present in front of us and a certain set of syndromes, certain conditions, it evokes other associations, the things we need to check on. And so as we can inform a greater understanding of these conditions, it can lead to improved screening and understanding of things people need to be watched out for if they have one of these variants.
Absolutely. Yeah, that's why that was extremely exciting just to think about that. And again, thinking back to just the principles of prevention and getting to some of these unmet cancers, let's say, a lot earlier when they're not cancer, pre-neoplastic is extremely important. So I take my hat off on that one. Another one, just that, you were just mentioning sort of conditions where there's, may not be a lot of genetic variance, or we might not have the strong associations yet. One of the ones you point out, this is a gastroenterology paper from 2021, Association of Genetic Variation with Cirrhosis, multi-trait genome-wide association gene environmental interaction study. And again, here, okay, you point out, okay, we don't have a lot of genetic variants, but obviously there's other things that contribute in terms of the way our genes interact, in this particular case with things like alcohol toxicity, the hepatotoxic effects of alcohol, and obesity. And here it comes in sort of the gene environment, gene, what do we want to call it, epigenetics or epigenome, whatever we would refer to, sort of everything else that is affecting the genome. Say a few words about this one too, because you mentioned the wearables and sort of some of the electronic connections to gathering this other data. Talk a little bit about this particular study, if you would.
So, the genomic associations with cirrhosis and all sorts of liver disease are such a complicated set of scenarios. And we know and long talked about things like alcohol use as an association. We know some hereditary conditions that can lead to liver disease, and often combinations of liver disease and other, such as lung disease. Yet, obviously there's complicated by, for instance, obesity and metabolic syndrome. There's an increasing incidence of these kinds of associations. And we also know things like viruses, like hepatitis C, especially hepatitis B, and co-infections with hepatitis D, all these environmental influences, as you mentioned. So from things you could be infected with to habits that you have to your genetics can play in a role in developing of liver disease. And cirrhosis obviously is a hugely impactful on life. And the fact of having inflammation puts you at risk for things like liver cancer as well. Yep. And so these are all really important things. And understanding genetic influences of these associations are helpful as we think about underlying risk. And this is a point where I'll point out, like, the importance again of things like All of Us, because we know there's differential risk of liver disease in different populations. And as we grow to include more diverse populations, it'll be really important for us to understand greater some of the roles that different disease risks might have in different populations and different genetic architectures. And that will help us define what those risks are, and think about the importance of, you mentioned screening in the cancer study, and screening is really important, obviously, as we think about liver disease as well. And if you have that kind of risk, I can think about a patient of mine who had underlying genetic risk for hereditary liver disease, lung liver disease. And, my advice to him was, really there's not a healthy level of alcohol use you can have, and I think that's part of the future of what we will be doing with genetically informed, personalized, or precision medicine. And I think these kinds of studies really point to where we're going to be going with those, the future of medicine.
Excellent. Absolutely. Josh, one more I'd like to ask about, and this was quite recent. This was back in July of this year, a paper in Human Genetics and Genomic Advances entitled Genetic and Clinical Determinants of Telomere Length. And, we the whole area of the gerosciences, Healthspan, compression of morbidity. These topics have been very hot for us in recent years. Obviously, you got the 2009 Nobel, the Blackburn, Grider, Shestak, telomerase work. And again, I'm very interested sort of to the types of things that you're looking at, finding out in terms of just aging, the biology of aging in general via the All of Us program. But what interestingly, I guess similar to the Nobel work that came out of your study here is that, sort of this Goldilocks phenomenon. Yeah, too long, no good; too short, no good. There's a common ground there that we should be aiming for when we're studying sort of these biomarkers, let's say, of a longevity and health span. What are you doing in this area, and what gets you excited in terms of sort of health span and sort of the gerosciences?
Yeah, no, great questions, and we all have a natural interest in that topic, don't we?
Yeah.
As we think about, I mean, telomeres have caught a lot of interest, and it's something that I guess we've had some interest. I've had some interest in it some time in thinking about from a variety of perspectives as we generate these clinical data sets. And of course, we're typically measuring telomeres from blood leukocytes, which have their own has its pluses and minuses. I mean, if you go through chemotherapy, obviously you cause there are environmental effects that distinctly will affect those through things like chemotherapy, which will create a big differential between your telomeres in the blood, for instance, versus potentially other places.
Mm-hmm.
That being said, those are environmental influences, and most people don't have strong necessarily environmental influences on their telomeres, like that kind of example. So in this study, again, it's applying it's the same rubric that you've been highlighting here about looking across the phenome, and so looking at measured telomere length and creating genetic predictors of that, and then looking at what the distribution of telomere length looks like and what it might be associated with. And you might think that it's just great to have a super long telomere because that protects you as you go through replications from their telomeres getting short. But what this study showed is that you see clinical associations with both short telomere length and long telomere length. You see a greater difference in lifespan with short, very short telomeres than very long telomeres. But in both cases, you have risks. And for having telomeres at the extremes of expected. And we, of course, saw a lot of the known genetic signals. We picked up a couple potentially new ones in there. And then we have a model that looks across the phenome and looks at some of the clinical associations we're seeing at both extremes. I think, what I'm excited about in the future, I think it's like many of these other spaces. There's a lot we don't know, and most of our data continue to be derived from, non-diverse populations. And so, we've seen in other conditions that it might matter why and what genes, for instance, might be driving a long telomere versus a short telomere. Mm-hmm. And as we get more data, it might matter, why you have a long telomere in terms of risk, for instance. Mm-hmm. So there might be some disentangling of some of those genetic signals that lead to something like that. As we look at diverse populations, we'll undoubtedly discover new associations with long, short telomeres and, the influence of environmental factors and genetics across that. Think about, too, the interactions of some of these. I mentioned the environmental exposures like chemotherapy and the effect. But, might there be interactions between those exposures and some potential protective factors of your telomere? And might those kind of interactions, make a difference in your resiliency, for instance, to taking chemotherapy? Mm-hmm. And so I think those kinds of questions, as we go further and deeper into these kinds of analyses, will be, where a lot of the future lies is really disentangling. And I think you could apply the same sorts of questions to cholesterol levels, all these, and that's been explored more obviously to date, but there'll be other things we uncover like this where we might find some really dramatic advances and discoveries. And I'll just highlight one thing. We talked about telomeres, a lot of interest there, but right now we have our first real telomere to telomere kind of understanding of the genome, and areas like the centromere that we've never mapped well.
Yeah.
And what's going to be buried in those areas that we haven't seen. And for once upon a time, if you go back a decade plus, there was all these areas of the genome that we just kind of considered dark matter and not necessarily very interesting. And yet that has all sorts of influences. And so, we might be next time we do this conversation, we might be really focused on some cool centromere aspect that no one knew existed.
Absolutely. Yeah, the dark genome, that's untapped. But again, a tremendous amount of data, no doubt, will pour out of that one as well, that you're going to have to crunch in your models. But, that being said, in March 2021, you and NIH director Francis Collins published this paper entitled "Precision Medicine in 2030: 7 Ways to Transform Healthcare." And there's a nice figure laid out, everything we've talked about already in terms of diversity and large cohorts and phenomics and EHR and so forth. And then there's the big section in the corner on big data and artificial intelligence. Would love to hear your story, your thoughts on where this is all going. And obviously you need the AI, the machine learning, and all these tools to crunch not just millions of EHR records, but everything else that's coming, whether it's the transcriptomics, the dark genome, everything else is going to be plugged in here. What is big data's role? What's AI's role here? And where do you see all this headed? Because obviously you need computer help when you're doing this work.
Certainly. I'm kind of glad we wrote that before ChatGPT came out. ChatGPT didn't write it.
Right.
The importance of big data and AI are really in the forefront of everyone's mind now, and I think it will just continue to grow. One of the things a study that actually was the first study that came out, at least in a digital form, out of using all of us data, which I think is really emblematic of some of a simple case of the potential, perhaps, and a really important case of how we need to have the right data underlying these models to do something important. And so in this study, the researchers started with a model developed at a single site, UC California at San Diego, to detect, who with glaucoma would need surgery. And they put all these features into the model, and they developed a model that worked very well. And then all of us comes out, and they think, well, let's apply this model on all of us, and I'm sure the idea was, we'll be able to show that our model replicates, and it's transferable, and it's applicable and generalizable. But it didn't. It did not work on all of us well at all, and in fact, its performance was near random chance. When they train a model on the all of us data, they recover incredible performance, very predictive models, just like they did within a single site. But the key difference is, there's more than 14, EHR vendors. There are, all the different kinds of insurers behind it. There's, diversity across the data in all aspects. People think about first, maybe the importance of diversity of things like racial and ethnic diversity, or having people who are rural and urban and exposures. But, there's another kind of diversity that's really important too. Like, did it come from Epic or Cerner? When you're thinking about electronic health records, because there are influences there that could actually also cause problems in how you've trained your model and how people enter in data. One of the very first algorithms I built, actually, decades ago, very immature sort of machine learning approach, but it uses support vector machines to identify people who had colorectal cancer, and we used full text notes to do it. And what we found, actually, the algorithm performed beautifully, and it should not have performed so well. And what we learned is every single person with colorectal cancer went through a certain one person, and they had their temperature encoded in their chart a very certain way that was completely distinct to having colon cancer. And it had nothing, it was nothing biologic, nothing clinical, but it was a formatting thing that the algorithm could learn beautifully. And so the algorithm that worked with almost 100% classification accuracy, it really would have meant nothing if we changed the form. And so that's just another example of, you got to have the right kind of training data coming into the resource. And yes, LLMs and other more modern approaches are getting much better at sort of quote, thinking through and being robust to these kinds of local minima. But if you don't have, kind of that diversity and broad data coming in across all the axes you need, you could learn things that, are not transferable and generalizable. That being said, I think it's an incredible tool, and we are building those resources that these things are really going to transform where we are. I mean, you see the studies, I'm sure, and your listeners will know of the studies. Recently saw stuff about, diagnosing clinical images. There's been stuff looking at case reports from New England Journal of Medicine, and how well these systems do, or taking tests, of course. There's lots of really exciting things, I think, on the horizon for what machine learning approaches can come behind us and help us with. So we have lots of great help with things like coding now, and generation is going to come into our lives, and I think we'll see the same thing in health, and maybe predict that, lots of information, predict the next drug in a way that maybe we can verify is an assistant in a good way for a physician to provide better care.
Mm-hmm. Mm-hmm. And moving a little bit from sort of the software and the sort of AI tools to some of the sort of the new or the hardware that's coming, because obviously, in the sort of the Josh Denny personalized precision medicine view of 2030, 2035, whatever, there's going to be other stuff happening. We've profiled the development of things like the digital twin, which, can you put the Iropaster over here and test out some different drug combinations before the actual Iropaster gets it? The distributed clinical trial that moves sort of the old school model of clinical development beyond just a few of these teaching hospitals to everyone through pharmacies and other types of networks. What else gets you excited in terms of the technologies that may be sort of in beta right now, but they're going to be a big part of what you envision here for this integrated, personalized precision future?
I think if I talk a little bit about all of us and as a paradigm, so all of us was always designed as a platform for lots of people to do research. And one of those kinds of things that I'm excited about is what we call ancillary studies. So that's building off of all of us to do other kinds of studies. One example, a big example of that right now is nutrition for precision health. So this is a study looking at, how can we be a lot smarter on nutrition. You could call it the most common drug we take, as we imbibe these exogenous compounds and have obviously dramatic influences on our health. And so how can we be more precise on that, integrate the genome, big machine learning, obviously opportunities in all parts of the spectrum. Can we be smarter about, cataloging what you're eating? But obviously, can we also be smart about recommending, maybe what makes sense for you in terms of a diet? And we don't think that's one size fits all. Just like we know that's really not for medicine, it's not for food either. And so that's an example. I think you mentioned clinical trials and ways we could do those differently. Doing distributed clinical trials is one part, or decentralization of them, but also what other kind of information can you glean from that? And so one example is can you set up a clinical trial, which we really don't do today, based on understanding of someone's genetics? I think the future is that you have these kinds of molecular information known for us and maybe, sequenced, maybe it becomes common to be sequenced at birth. Maybe it, is later in life, but having the sequence read once, use many times, kind of understanding will, portend to pharmacogenomics and the right drug at the right time. It'll also, though, I think, help us think about, clinical trials that are based on genetic risk and maybe faster, simpler clinical trials on drugs, because we'll be able to really think about the distribution of one's risks across not just clinical factors, but also maybe genetic factors and targeting of those kinds of things in a positive way to get quicker answers, more precise answers, and move towards precision medicine in those regards. So I'm really excited about that. And all of us is one platform that can launch those. I think in the future, a lot more of us will have genetic data, and we'll have electronic health records that are smart enough to think about genetics and incorporate that and sort of have more people in and offer them tailored clinical trials as well. But it's going to start with programs like All of Us, and hopefully we create the value proposition that this moves and advances medicine faster.
Mm-hmm. I love it. Really love it. And I appreciate your connection there prior to sort of the nutrigenomics space. And as you were saying, all the, the 99% of all the other stuff that we take beyond medicines, like our nutritional products, our foods, that, these are bioactive substances that do things that, I really appreciate all this. It's really an amazing time for this program. What's coming up? I know that, I think just the other day, the American Medical Informatics Association annual meeting just happened. I think you were represented there. Anything else that we should know about coming up for the rest of, we're almost at the end of 2023 now, but as we get into 2024, any new initiatives for the program, or conferences, talks, other places that we can follow you, run into you, possibly meet you, and listen to what you're doing? Anything that I missed, please take the floor.
Yeah, thank you. Well, the biggest thing we have right now that we're working on is pediatrics. Okay. We have always wanted to be inclusive of the lifespan. Right now we're 18 and over. Because we have electronic health records, we have people that have shared their pediatric electronic health records in time. But we aren't recruiting children, and so we have been working hard this year on readying launch of a pediatric program that will include enrolling people 0 to 18, and that is something that will start just like we'll start with very small tests, see what we need to refine, and then move up. And we'll start with the youngest individuals at select locations as we test this program, and then we'll be revising and scaling it up over the next couple of years. And that will be in partnership, of course, with our adult program. So that's one thing. We're pretty much at the beginning stages of our ancillary study program, and I'm looking forward to bringing out more of that in our program. And so we've had partnerships with other institutes and centers across the NIH, which are developing. Nutrition Precision Health has actually 18 institutes, centers, and offices as part of it. So that's a really broad collaboration across the NIH. We're talking with National Eye Institute, National Heart, Lung, and Blood Institute, National Human Genome Research Institute, and lots of other ICs across this as different partnerships in this process. And what we could do to continue to advance this is a platform for understanding of human health and disease quite broadly. And so I will continue to have our footprint at lots of conferences and continue to grow that. It's so exciting to see the researchers. It's just really explode. We've more than doubled our researchers in 2023, and I could say that statement in September. So it just keeps extending, and so those researchers are doing lots of things and writing important papers. And there's just a bunch of stuff that came out at the AHA. This isn't published yet as presentations, but it's hitting a lot of press about the influences of marijuana on heart disease and coming out of our program. And it's exciting to see, sort of the use of these data in ways and thinking about what are contemporary changes in legal practices that could rate change in exposures and create new hypotheses for study, as well as provide evidence to help us think about public health issues, as well as the facts like genomics and precision nutrition. All those things put together, I think we have lots of use cases for our program that we broadly want to support.
It's going to be an exciting few coming years. I followed you in the program for the last couple of years. Really enjoyed that we were able to get together and do this now, and we'll be happy to do a follow-up as these amazing data sets come in, and the new findings they're providing. Just really amazing work. I take my hat off to you. For everybody that is going to be listening to this particular episode of our show across the various podcast networks or watching on the YouTube channel. Again, you've been listening to Dr. Josh Denny, Chief Executive Officer, National Institutes of Health, All of Us Research Program, doing a really amazing things to accelerate precision, personalized medicine for all populations. Josh, again, I want to thank you for taking the time out of your schedule to come talk to us for a little while, educate us on what you're up to. Obviously, thank you for what you do for all of us here in the United States. And it's like you like to say on our show, thanks for helping to create a better tomorrow for everybody via what you do. A really, really great story, and wishing you best with the program.
Ira, thank you so much. It's such a pleasure to talk with you, and appreciate the time.
Great having you.